Chemical evolution suggests that the Earth's early atmosphere, under reducing conditions, allowed organic molecules to form spontaneously. Serpentinization is a geochemical reaction in hydrothermal systems that produces H2 and fuels microbial metabolism. Differences between eukaryotic and prokaryotic cells. Prokaryotic cells lack a nucleus or organelles, have circular chromosomes, and divide by binary fission. Eukaryotic cells have a nucleus with linear chromosomes, organelles like mitochondria, and cytoskeletons. They reproduce through mitosis and have polysaccharide cell walls. All cells use DNA to store genetic information and ATP for energy. Animal cells lack a cell wall and have a flexible membrane. They contain organelles like lysosomes. Plant cells have a rigid cell wall made of cellulose, chloroplasts for photosynthesis, and large central vacuoles. Bacterial cells have a simpler structure without membrane-bound organelles, and their cell walls are made of peptidoglycan in bacteria or pseudomurine in archaea. The generation of ATP likely evolved in 3 stages. Glycolysis, an ancient and universal pathway that does not require oxygen. Photosynthesis evolved later, allowing cells to harness sunlight for energy. Oxidative metabolism developed after the Great Oxygenation Event, using oxygen for efficient ATP production. 4 major families of small organic molecules. Sugars, energy, structural roles. Fatty acids, membrane structure, energy storage. Amino acids, protein building. Nucleotides, genetic material, energy carriers. Monomers forming macromolecules. Sugars, polysaccharides. Fatty acids, lipids. Amino acids, proteins. Nucleotides, nucleic acids. RNA and DNA. Deoxyribose, ATCG, double helix, stores genetic info. RNA, ribose, CG, single-stranded, protein synthesis. Gregor-Mendel. Mendel's pea plant experiments established the principles of inheritance. Archibald Garrod. Garrod studied alkaptonuria, brown piss, and proposed the 1-gene-1 enzyme. Beadle and Tatum. Using Neurospora, fungus, they confirmed the 1-gene-1 enzyme. Frederick Griffith. Griffith discovered the transforming principle through his experiments with Streptococcus pneumoniae. Avery, McCarty, and MacLeod. They identified DNA as the transforming principle in Griffith's experiment. Hershey Chase. Using bacteriophages, they confirmed that DNA, not protein, is the genetic material passed from viruses to bacteria during infection by labeling DNA with radioactive phosphorus and proteins with radioactive sulfur.